The Center for Dryland Agriculture is a regional center. We have students from all over Africa, from the west coast to the east coast of Africa. We have graduate students coming here to study. We also have other students coming on internships to use our facilities. We have, as you know, a research farm that has very nice facilities for irrigation, fertigation, high-value crop production, nurseries for raising seedlings, and so on. We also have a number of facilities and laboratories. One is the tissue culture laboratory, where we do research on propagation of different plant materials. We also have other laboratories, such as the molecular biology laboratory, where we do DNA extraction and other activities involving molecular biology. We have a general laboratory where we do soil analysis, fertilizer analysis, analysis of pesticides, and so on. We have a food analysis laboratory where we determine the protein content, fruit fiber, oil, and so on of different food and feeds. We have a GIS laboratory where we do geographical and spatial analysis, remote sensing, and so on.
The change of rain pattern, drought, low soil fertility, desertification, and low yield, as well as extinction of some medicinal species, are a few of the unnumbered complicated problems that threaten food production and security in dryland areas of sub-Saharan Africa, leaving millions of lives in danger. The heart of development and backbone in today's modern world we see in the developed countries is research. However, research institutions in Africa lack the required sophisticated apparatus used in conducting modern scientific research. Interestingly, this story is changing now gradually with the support of the World Bank and other partners.
The new equipment we just acquired, I think this should be the first one to best of my knowledge in this country, the mid-near infrared. And then with this collaboration with the crop nut people, I think it will be very, very good for farmers in Nigeria and even beyond Nigeria. As soon as you introduce your soil sample, it will be able to even recommend the fertilizer you're supposed to use, the type of fertilizer, and even the quantity of fertilizer you're supposed to use per hectare.
In the heart of northwestern state of Kano, Nigeria, inside the Bayer University Kano, a unique, modern and vibrant research training center named Center for Dryland Agriculture, CDA, is well equipped with sophisticated laboratories and practical field farms with the primary aim of bringing solutions to dryland agricultural problems in Sahelian countries of Africa through research and innovations. Join us to take you inside the research and training facilities in the laboratories and the farm of the CDA. All this is for you to know the types of laboratories the CDA has and understand the functions of the modern apparatus inside the laboratories, how to access their services, and the breakthrough researches conducted. We will as well take you to CDA Research and Training Farm to see and test hybrid vegetables and sweet fruits grown in greenhouse and open field. Are you not drooling? We won't blame you. They are fresh from the farm that looks succulent.
Let's talk about the recent research that we conducted about that palm. You see, the molecular biology created this avenue where we can be able to tell the sex of a particular plant at an early stage.
Let's begin taking you around from the laboratories. If you check the package of food items you purchase from stores, you'll see a table that contains nutritional information about the product. This information comes from analysis conducted in a laboratory called Food and Feed Laboratory. The Center for Dryland Agriculture, CDA, has this type of laboratory equipped with automated machines that reduce human error in the analysis.
The lab is equipped with modern equipment. We have most importantly, an proximate parameter called protein. Protein is a very important parameter in proximate analysis, and it tells a lot on how nutritious the food is. We have an equipment that measures that. It's called the GelTAC equipment. It is an automatic equipment that distillate the sample, titrate it, and then display the protein content instantly on the screen for the analyst to know.
The proximate parameters analysis conducted in this laboratory include energy, protein, moisture, crude fat, fiber, and ash parameter. These analyses ensure the quality of human and animal food and help consumers know what they are consuming.
In this university, as far as food quality is concerned, this is the most sophisticated lab we have in the university. I don't know the other universities, but hardly you go to any nutrition laboratory and find equipment like this. They are all state of the art. They are all automatic. When I say automatic, you reduce a lot of human intervention in your analysis, thereby reducing the chances of error. Whenever you want to do things classically, there are so many sources of error. But when you have a precision equipment that is calibrated, then the chances of introducing error in your analysis is reduced.
Malam Hashim Eyakase took us go around the laboratory and explained the functions of the automated machines installed in this laboratory.
This is where you put your samples from here. You see your sample coming out. You got it, Ba? Yeah. You got it? Yeah. Okay, so this is now your sample. If it requires that you sieve into a more finer material, you sieve depending on what you are going to do. Yes.
This laboratory is very unique in the outlook of the apparatus, and that may be the reason why researchers from West and Central African countries are trooping in to conduct their research in conducive atmosphere here at CDA BUK.
CDA is very unique, I must tell you. You will attest to what I'm saying, that this lab is highly sophisticated. And we have a lot of PhD students. We even have international students. I have worked with them coming to this lab to work. So the environment is very rich in terms of the resources we need for molecular research.
Indeed, the standard of these lab services is attested by individuals and companies that brought different samples for analysis. They have a story to tell about the efficiency of this laboratory. At the adjacent of the Food and Feed Laboratory is a molecular biology laboratory, which is very important in solving numerous agricultural problems that change the development of modern agriculture in Sahelian countries. One of the breakthrough researches conducted in this laboratory was the sex determination of the date palm plant. Before this research, date palm farmers lose a lot, but now the story has changed. Hassan S. Khalid is a chief technologist in this lab and is very proud of this research and several others.
I am happy to be here. Actually, the research that I'm most happy to be part of is the sex determination of the date palm tree, which is a huge one. I think it's been sponsored by government of Jigawa State, where we identify the sex of that particular plant at the very early stage, which we provide 100,000 of seedlings to the government. I think that's a very, very big one that's been conducted in this very laboratory. And we have researchers, probably MSc students and PhD students, who actually work with resistant genes, stress genes. These are researches that I'm very proud of that are being conducted here. Just recently, about 2 weeks ago, there is this very research about checking the yellow stripe from sorghum. And this yellow stripe aids in their iron transportation. So, which is being conducted here in this center. I'm most proud of it because the lady that I worked with actually did a very good one.
Other analyses conducted in this laboratory are DNA extraction, RNA extraction, protein determination, polymerase chain reaction, nanospectrometry, microplate spectrometry, microbial culture, cell count, gram documentation, and bioinformatic analysis, Hassan S. Khalid talked to us about the digital services they use to conduct these analyses.
I'm really confident with our staff here. I'm really confident with it because most of them are my colleagues. We are technical staff here. We conducted the work. Even if there is error, we normally go back to the work and check. We have our Deputy Director of Research, which is also a molecular biologist, and he normally oversees all activities to make sure everything is implemented well. And again, we have a coordinating technologies, which normally give us the result. We don't give out the result directly. The coordinating technologies check the result, verify it, validate it before putting it out.
We met Muhammad Nuruddin, a master's student from Department of Plant Biology, who is conducting his research on diseases related to cowpea.
And by the end of this research, we will be able to identify those resistant genes in those cowpea. Now, if like I'm working with 44 varieties, I was able to identify 5 varieties that are resistant to these cowpea mosaic virus. Now, to do this in the field is going to take me much number of years because I have to plant again and plant so that I will be sure that these varieties are resistant. But right now, with the advent of molecular biology, I will just develop my primer, send it for to Inkaba in South Africa, and with this primer I will be able to identify these genes. Now, if these genes are present, it means the varieties are resistant, and if they are absent, it means the varieties are susceptible.
We have 2 more laboratories to visit, but before then, let's go to the CDA training and research farm to eat some fruits and take fresh air. The CDA farm is a practical learning field for researchers and students, and a commercial unit expected to fund its activities in the near future. As our team approached the farm, we heard the sounds of the lectures coming from the greenhouse. It was students from Botany Department, Ahmed Bello University, Zaria, who came for a field trip course taking lectures in very practical question and answer sessions. It was quite amazing.
What is the ratio of concentration between the fertilizer and the water?
Fertigation, as I told you, is a process that we mix water and fertilizer. Now, we have, if you are adding urea to maybe 20 liters of water, what you need is just 50 grams of urea. 50 grams of urea in 20 liters of water. So our tanks are graduated. From there we calculate if we are giving 100 liters or 200 liters, then we calculate based on that formula.
The team was led by their head of department, Professor Bashir Yusuf Abubakar, who told us they came to CDA to expose the students to some practical skills. He was amazed with the kind of apparatus the laboratories has, despite being from the ABU, which was the